Hello everyone, this is Payal Vaja. Welcome to Pharma Desert. In this video, we will discuss about classification, reproduction and cultivation of viruses. So let's get started. These are the list of contents we will discuss in this video. First one is introduction, next one is viral structure, next one is classification of virus, cultivation of virus and reproduction of virus. We will discuss one by one. First one, introduction. Viruses were originally distinguished from other infectious agents because they are especially small or filterable and because they are obligatory intracellular parasites, that is, they absolutely require living host cells in order to multiply. So what is viruses? Viruses were originally distinguished from other infectious agent. Why viruses that are distinguished or differentiate from other infectious agent? Because they are specially small and because they are obligatory intracellular parasite and they absolutely require living host cells in order to multiply. So viruses that are differentiated from other infectious agent. Why? That is required because they are smaller in size as well as they are obligatory intracellular parasite and they absolutely require living host cell for multiplication. Once viruses enter a host cell, the viral nucleic acid becomes active and viral multiplication result. So how viral multiplication that is possible? Once virus that is enter into a host cell, viral nucleic acid, it becomes activate and viral multiplication occur. In this sense, viruses are alive. When they multiply in the host cell, they infect. From a clinical point of view, viruses can be considered alive because they causes infection and disease, just as pathogenic bacteria, fungi, and protozoa do. So, in this sense, viruses that are alive and when they multiply in the host cell, they infect. And from a clinical point of view, viruses which can be considered as alive because they causes infection as well as they causes disease and just as a pathogenic bacteria, fungi as well as protozoa do it. What is virus structure? The structure of virus are as below. So virus that are made up of virion, capsid and envelope, nucleic acid, either made up of DNA or RNA, capsomers and nucleocapsid. First one, a viron or a virion. It is a complete, fully developed infectious viral particle composed of nucleic acid and surrounded by a protein coat that protects it from the environment and is a vehicle of transmission from one host cell to another. So, what is virion? It is a complete, fully developed infectious virus particle and they are made up of nucleic acid surrounded by protein coat and they protect it from environment and that is a vehicle of transmission from host cell to another cell. So virion, that is a complete, fully developed infectious virus particle. They are made up of nucleic acid and that is surrounded by protein coat that protect it from the environment and it is a vehicle of transmission from one host cell to another one. Next one is nucleic acid. A virus can have either DNA or RNA but never both. So virus that is made up of DNA, otherwise that is made up of RNA. But they do not contain both DNA as well as RNA. The nucleic acid of virus that can be single stranded or double stranded. So nucleic acid of virus that is made up of single stranded DNA or double stranded DNA. Thus there are viruses with the familiar double stranded DNA, with single stranded DNA, with double stranded RNA and with single stranded RNA. So viruses, they are made up of double strand DNA with single stranded DNA. Otherwise, they are made up of double stranded RNA with single stranded RNA. Depending on the virus, nucleic acid can be a linear or a circular in nature. In some viruses, such as influenza virus, the nucleic acid is in several separate segments. That is the figure of enveloped virus. First one is in center portion, capsomer, that is the red in color, capsomer that is present circular red in color. In capsomer inner side, nucleic acid is present, that is purple in color. Above or outside the capsomer, envelope that is present, that is green in color. And last one is capsid that is present, that is blue in color, and that is entire protein coat. That is the figure of enveloped virus. Next one is capsid as well as envelope. The nucleic acid of virus that is protected by a protein coat that are called as capsid. We already see what is capsid in the last figure or last slide. Nucleic acid of virus that is protected with the help of protein coat, and this protein coat that is called as capsid. Each capsid that is composed of protein subunit. These protein subunit that are called as capsomers. So blue colors, various capsomers is present. Individual capsomers are often visible in electron micrograph. And in some viruses, capsid that is covered by envelope, which usually consists of some combination of lipid, protein and carbohydrates. First one, what is capsid as well as envelope? So capsid, nucleic acid of virus that is protected with protein coat that are called as capsid. While capsid, they are divided into various protein subunits that are called as capsomers, and individual capsomers that are visible with the help of electron micrographs. And last one is what is envelope. In some viruses, capsid that is covered by an envelope and which usually consists of some combination of lipid, protein and carbohydrates. So that is the figure A, that is the polyhedral virus. In polyhedral virus, capsid that is present and subunits that are called as capsomers, and in capsomers nucleic acid is present in filament form. In another side, mastadinovirus that is present, diameter that is about 30 nanometer. Depending on the virus, envelopes may or may not be covered by spikes, which are carbohydrate protein complexes that projects from the surface of the envelope. And some viruses attach to the host cell by means of spikes. So depending on the viruses, envelopes, it may or may not be covered by spikes, which are carbohydrate protein complex that project from surface of envelope. And some viruses, they are attached to the host cell by means of spikes. Spikes are such a reliable characteristics of some viruses. What is spikes? First one is an enveloped helical virus. In enveloped helical virus, first one is spikes that are present above the envelope that are green in color, spikes that is present. Second one is envelope that is present that are yellow in color. Below the envelope, blue color nucleic acid is present in a filament form, and last one is capsomer that is present. that is also blue in color, whilst figure B, spikes that is present on influenza virus and diameter that is about 50 nanometer in diameter. That is the schematic diagram of influenza virus, and that is connected or conducted with the help of TAME study, transmission electron microscopy. Next one is classification of viruses. Viruses that may be classified into several different morphological types on the basis of their capsid architecture. So viruses, they are classified into several different morphological types basis of their capsid architectures. The structure of this capsid has been revealed by electron microscopy and a technique that are called as X-ray crystallography. So structure of this capsid that is revealed with the help of electron microscopy and this technique that are also called as X-ray crystallography. Classification of virus. First one is helical virus, next one is polyhedral virus, next one is enveloped virus, and last one is complex virus. First one is helical virus. What is helical virus? Helical virus resembles long rods that may be rigid or flexible in nature. So helical virus, they are made up of long rods. They are long rods that may be rigid in nature or they are flexible in nature. The viral nucleic acid is found within a hollow cylindrical capsid that has a helical structure. So nucleic acid that is found within a hollow cylindrical capsid and that have one type of helical structure. And the viruses that cause rabies and Ebola hemorrhagic fever are a helical viruses. So what is helical viruses? They are look like a long rod shape. They may be rigid in nature or flexible in nature. The viral nucleic acid that is found within a hollow cylindrical capsid. So nucleic acid that are found in hollow cylindrical capsid and that has a helical like structure. Viruses that causes rabies and Ebola hemorrhagic fever that are helical virus. Next one is polyhedral virus. What is polyhedral virus? Many animal, plant and bacterial viruses that are polyhedral in nature or many sided viruses. The capsid of most polyhedral viruses is in the shape of an icosahedron, a regular polyhedron with 20 triangular faces and 12 corners. So polyhedral virus that are present in many animals, plant, bacterial viruses and capsid of most polyhedral virus is in the shape of icosahedron and a regular polyhedron with 20 triangular face and 12 corners are present. So regular polyhedron that are made up of 20 triangular face and 12 corners. The capsomers of each face form an equilateral triangle and an example of polyhedral virus in the shape of icosahedron is the adenovirus and another icosahedron virus is the poliovirus. So, capsomer of each face, they form a equilateral triangle as well as example of polyhedral virus in the shape of icosahedron is the adenovirus and another icosahedron virus that is the poliovirus. Last one is enveloped virus. Capsid of some virus that is covered by envelope that are called as enveloped virus and enveloped virus that are roughly spherical in shape. And when helical or polyhedral viruses are enclosed by envelopes, they are called as enveloped helical or enveloped polyhedral viruses. Example of enveloped helical virus, that is the influenza virus. So what is enveloped virus? According to their name, enveloped virus, that is the made up of or that are enclosed by envelopes. So capsid of some viruses that is covered with the help of envelope, and envelope viruses that are roughly spherical in shape. When helical or polyhedral viruses that are enclosed by envelopes, they are called as enveloped helical or they are called as enveloped polyhedral viruses. Example of enveloped helical virus, that is the influenza virus. First figure is helical virus. So helical virus that are made up of capsid and capsid that are divided into various subunits that are called as capsomers, and in capsomers hollow space is present, in hollow space nucleic acid is present. Second one is polyhedral virus. In polyhedral virus capsid is present. In capsid there is the one type of triangle like structure. In triangle like structure nucleic acid is present in filament form as well as capsomers is present. While last one is enveloped helical virus. In enveloped helical virus, spikes is present above the envelope, that is green in color, and envelope that is present, that is yellow in color. Below the envelope, nucleic acid is present, that is blue in color, that is filamental in nature. And last one is capsomer is present, that is also blue in color. Next one is complex virus. Some virus, particularly bacterial virus, have a complicated structures and are called as complex virus. So what is complex virus? Any virus that have a complicated structures that are called as complex virus. One example of complex virus is a bacteriophage. So bacteriophage that are called as complex virus. Some bacteriophages have a capsid to which additional structures are attached. So some bacteriophage they contain the capsid and to which additional structures are attached. In this figure, notice that the capsid head is polyhedral and that the tail sheath it is a helical and the head contains nucleic acid. So we will see the figure and notice that capsid or head that is the polyhedral in nature and tail sheath that is the helical and head that contain nucleic acid. That is the figure of bacteriophage. In bacteriophage capsid that is the head that is 65 nanometer in diameter. In capsid DNA that is present, purple color DNA that is present. Below the capsid, sheath that is available and tail fiber is present. Pin that is available and last one is base plate is available for sheath. That is the T1 bacteriophage. And that is the figure of second one is microscopic view of bacteriophage that is 40 nanometer in diameter. Next one is cultivation of viruses. First one is growing bacteriophages in the laboratory. Bacteriophages which can grow either in suspension of bacteria, in liquid media or in bacterial culture on solid media. So bacteriophage which can be grow either in suspension of bacteria in liquid media, otherwise it can be grow either in bacterial culture on solid media. So bacteriophage they are grow with by two situations. First one that is grow with the help of suspension of bacteria in liquid media. Otherwise they will grow in bacterial culture on solid media. A sample of bacteriophage that is mixed with host bacteria and melted agar. So sample of bacteriophage that is mixed with host bacteria and melted agar. Agar which containing the bacteriophages and host bacteria is then poured into a petri plate containing a hardened layer of agar growth medium. So agar which containing bacteriophages as well as host bacteria, and that is pour into a petri plate, and this petri plate they contain hardened layer of agar growth medium. So that is regarding growing bacteriophage in laboratory. So how bacteriophage they grow in the laboratory? First one, bacteriophage can be grow either suspension of bacteria in liquid media, otherwise they grow in bacterial culture on solid media. In the sample of bacteriophage that is mixed with host bacteria and melted agar. After that, agar containing bacteriophage as well as host bacteria, they are poured into the petri plate containing hardened layer of agar growth medium. Each virus infects a bacterium, multiplies and releases several hundred new viruses. So each virus they infects bacteria, they multiplies and release several hundred new virus. These newly produced viruses which infect other bacteria in the immediate vicinity, and more new viruses are produced. So each virus they infect the bacteria, they multiplies and they release several hundred new viruses. And newly produced virus they infects other bacteria in the immediate vicinity, and more new viruses that are produced. Following several viral multiplication cycles, all the bacteria in the area surrounding the original viruses that are destroyed. While the plaque form, uninfected bacteria elsewhere in the petri plate multiply rapidly and they produce a turbid background. So while plaque are formed, uninfected bacteria elsewhere in the petri plate multiplies rapidly and they produce a turbid background. Each plaque theoretically corresponds to a single virus in the initial suspension. Therefore, the concentration of viral suspensions measured by the number of plaques, they are usually given in terms of plaque forming units. So, what is plaque forming units or PFU? Each plaque theoretically corresponds to a single virus in the initial suspension, because the concentration of viral suspension that is measured by number of plaques that are usually given in term of plaque forming units. That is the figure of, or that is the schematic diagram of quantification of bacterial viruses by plaque assay. First one is phage dilution. Second one is molted top agar, that is yellow in color, and pink in color, that is the bacteria cell that is pink in color. So molten agar that is transfer into a pour mixture onto the nutrient agar plate. So molted agar that is transfer into the nutrient agar plate, and sandwich of top agar as well as nutrient agar that is present. Top agar that is pour into the onto the nutrient agar plate. So sandwich that is formed on top agar as well as nutrient agar and incubate for some time period, and phage bacterial phage plate that is formed. So that is the quantification of bacterial virus with the help of plaque assay. First one, phage dilution is occur. Molten top agar that is present, that is yellow in color. Bacterial cell that is pink in color. Next one, molten top agar that are pour onto the nutrient agar plate. So nutrient agar as well as top agar form a sandwich and they are incubated for some time period, and phage plaques that is formed. Second one is growing animal viruses in the laboratory. How it is possible? Three methods are commonly used for culturing the animal viruses in the laboratory. First one is living animals, next one is embryonated eggs, and next one is cell cultures. First one, in living animals, some animal viruses can be cultured only in living animals such as mice, rabbits and guinea pigs. So some animal viruses they are cultured in Some living animals like mice, rabbit and guinea pigs. Most experiments to study the immune system's response to viral infections must also be performed in virally infected live animals. So most experiments to study the immune system response for viral infections, it should be performed in virally infected live animals. Animal inoculation may be used as a diagnostic procedure for identifying and isolating a virus from a clinical specimen. So why in animal inoculation that is required? Animal inoculation which may be used as a diagnostic procedure for identifying as well as isolating a virus from clinical specimen. After animal is inoculated with the specimen, animal is observed for a signs of disease. So when animals that is inoculated with the specimen, animals that is observed for signs of disease. AIDS vaccine are presently being tested in the humans, but the disease progress so slowly in humans that it can take years to determine the effectiveness of this vaccine. So AIDS vaccine that are presently being tested in humans, but disease progress so slowly in the human beings and that it can take years to determine the effectiveness of this vaccine. So AIDS vaccine that are presently tested in various human beings, but disease progress very slowly in human beings. So they can take years to determine its effectiveness for vaccines. These diseases are caused by lentiviruses and which are closely related to HIV, and the disease developed within a few months, thus providing a model for studying a viral growth in different tissue. So these diseases that are caused by one type of lentivirus and that are closely related to HIV, and the disease developed within a few months, they providing a model for studying a viral growth in different tissues. Next one is in embryonated eggs. What is embryonated eggs? If the virus will grow in an embryonated eggs, this can be a fairly convenient and inexpensive form of host for many animal viruses. So in embryonated eggs, if the virus they will grow in embryonated egg, this can be a fairly convenient as well as inexpensive form of host for many animal virus. So if virus they will be grow in embryonated eggs, they can be a fairly convenient as well as inexpensive form of host for many animal viruses. A hole is drilled in the shell of embryonated egg and a viral suspension or a suspected viral containing tissue that is injected into a fluid of egg. So, one hole that is drilled into the shell of embryonated eggs and a viral suspension or a suspected virus containing tissue that is injected into the fluid of eggs. Viral growth is signaled by the death of embryo, by embryo, cell damage, or by the formation of typical pokes or lesions on the egg membranes. So, viral growth that is signaled by death of embryo and by embryo cell damage or by formation of typical pokes or lesion on the egg membranes. So, how it is identify if the viral growth that is occur or not? So viral growth that is identified by the death of embryo, by embryo cell damage, or by the formation of typical pokes or lesions on the egg membranes. This method was once the most widely used method of viral isolation and growth, and it is still used to grow viruses for some vaccines. That is the inoculation of embryonated eggs. The injection site determines the membrane on which viruses will grow. So first one is air sac is present. In above portion air sac is present. After air sac shell is present. Amniotic cavity is available. In amniotic cavity, after amniotic cavity, chorioalanotic membrane is present. Chorioalanotic membrane inoculation with the help of injection. After inoculation, amniotic inoculation, alanotic inoculation, yolk sac inoculation. After yolk sac inoculation, alanotic cavity is formed, albumin is present and shell membrane is available. So that is the figure of inoculation of embryonated eggs. Next one is cell cultures. Cell cultures have replaced embryonated eggs as the preferred type of growth medium for many viruses. So cell culture that is replaced embryonated eggs as the preferred type of growth medium for many viruses. And cell cultures consist of cells grow in the culture media in the laboratory. So cell culture consist of cells that are grow in culture media in the laboratory. Because these culture media are generally rather homogeneous collection of cells and can be propagated and handled much like bacterial culture, they are more convenient to work with than whole animals or embryonated eggs. So because these culture are generally rather homogeneous collection of cells and they can be propagated and handled much like bacterial culture. They are more convenient to work with whole animals or embryonated eggs. So that is regarding cell culture. Cell culture lines are started by treating a slice of animal tissue with enzyme that separate the individual cell. First one is cell culture. Transformed cells, transformed cells can be grow identified in laboratory culture. First step a tissue that is treated with enzymes to separate the cell. Second one is cells are suspended in the culture medium. And third one is normal cells or primary cells grow in the monolayer across the glass or a plastic container, and transformed cell or a continuous cell culture do not grow in a monolayer. So that is the figure of cell culture. Next one, reproduction of virus. Reproduction of viruses. First one is T1 bacteriophages. Various stages is available for reproduction of virus. First one is attachment. Next one is penetration, biosynthesis, maturation and release. That is the life cycle of T cell bacteriophage. First one or first figure is bacterial cell wall is present, and in bacterial cell wall bacterial chromosome is present. First step is attachment. In attachment bacteriophage attaches to the host cell. Second one is capsidor DNA that is present. Second one is penetration. So first step is bacteriophage that is attached to the host cell. Second stage is penetration. So bacteriophage penetrate into the host cell. In penetration, bacteriophage penetrate host cell and injects its DNA. So we will see in the figure easily. We can see in the figure easily. Bacteriophage penetrate into the host cell and they release the DNA into the host cell. Purple colored DNA that is shown in the figure. In third stage, biosynthesis. In biosynthesis, bacteriophage DNA direct synthesis of viral components by the host cell. So in biosynthesis, various new viruses are formed. Next one is in maturation. In maturation, viral components are assembled into the virions and they are matured. Next one is after maturation, release, host cell, lysis or breakdown of host cell and new virions are released from the host cell. That is the figure of lytic cycle of T1 bacteriophage. If any query or doubts related to topic, please write your questions in comment box. Thank you.